Good morning, everyone, and thank you for joining us today for the NCI CBIIT Speaker Series. I'm Tony Kurlavich, the head of the Cancer Informatics Program at CBIIT. First, I want to remind everyone that today's presentation is being recorded and will be available on the wiki for the Speaker Series as a screencast with voiceover, and also posted on the Speaker Series YouTube playlist. And if you Google NCI Speaker Series, you'll find that wiki page. Information about this and future speakers is available on our website, so you can just Google CBIIT website. It'll be the first search result. And announcements also are available on Twitter. Our handle is @NCI_NCIP. Today we're very happy to welcome Dr. Jianxin Shi, a tenure-track investigator in the Biostatistics branch of NCI's Division of Cancer Epidemiology and Genetics. Dr. Shi received a PhD in statistics from Stanford University in 2006. He then pursued a postdoctoral research fellowship at the Stanford University School of Medicine, focusing his studies on the genetic mapping of psychiatric disorders from 2006 to 2009. The title of his presentation today is Characterizing the Genetic Basis of Methylome Diversity in Histological Normal Human Lung Tissue. And with that, I'll turn over the floor to Dr. Shi.
Thank you for the very nice introduction, and thank you for inviting me to present my work here. Let me first briefly introduce our branch here. The Biostatistics branch in the DCEG has now 8 tenured PIs and 4 tenure-track PIs. We also have 7 fellows and 1 staff scientist. Our branch has focused on the research in 3 main areas: in the descriptive epidemiology research and analytical studies, and also translational research related with vaccine trials and cancer risk modeling and evaluation of the new biomarkers. So we have been extensively collaborating with both intramural and extramural programs. In particular, we have been collaborating with all of the branches within the DCEG. So we are developing methodology to pushing forward all of these cutting-edge studies. So our methodology studies ranges from the standard EPI study, for example, the missing data analysis, measurement error, data quality control, to the cutting-edge new technologies, including microbiomics and cancer genomics. So today I'm going to talk about a genome-wide genetic mapping of DNA methylation in human lung tissue. So it's well known that the DNA methylation plays a central role in epigenetic gene regulation and disease susceptibility. Also, the methylation levels vary greatly across different samples. So such variation could be caused by both genetic factors and environmental factors. So methylation quantitative trait loci analysis, or the cis-meQTL analysis, aims to identify genetic variants associated with DNA methylation. So here's one such example. For this CpG probe, the methylation levels differ substantially across 3 different genotypes. So this is one example of methylation QTL. The cis-meQTL can be classified into 2 categories. We call this as cis if the SNP and the targeting CpG sites are very close, or trans if the CpG site and the SNPs are far away, or they are located on different chromosomes. So we are going to explore the next few scientific questions in this talk. We're going to investigate the extent and the pattern of genetic regulation of DNA methylation, and the existence of the master regulator, and also the heterogeneity of genetic control of DNA methylation across different tissues. Then we're going to investigate whether the methylation QTL data are useful for the GWAS studies of the lung cancer. Okay. And also we are going to discuss some of the analytic and computational challenges in such a big data analysis. So for this study, we have used 210 tumor adjacent normal fresh frozen tissues in the EAGLE study led by Dr. Landy and Caparoso here. First of all, we performed a principal component analysis to confirm that these normal samples are separate from the tumor samples. And then we used the new Illumina human methylation 450 platform to profile the genome-wide methylation. This platform has provided much more details compared to the previous generation of the platform. In particular, we can see that this platform covers almost all of the categories of the genomic regions. So for this analysis, for the cis-methylation QTL analysis, for each targeted CpG site, we have to search SNPs locally. So the computational complexity is the number of the CpG sites multiplied by the number of SNPs in the local region. Here it's roughly about 200 SNPs in this local region. But for search for trans associations for CpG sites, we have to search the genome-wide for all of the SNPs. So now the computational complexity is the total number of the CpG sites multiplied by the total number of genome-wide SNPs. So again, one more thing is that we need to run permutations to appropriately adjust for multiple testing. So as you can see, the computational burden is prohibitive. Because of this extremely high computational burden, trans-association analysis is rarely done for this high-dimensional methylation QTL studies. So we are going to use the NIH computational cluster BioWolf together with our statistical innovations. We were able to finish this trans analysis and review some important findings based on this analysis. So as we mentioned, for the cis analysis, we search for SNPs in the local region. Here we use the 500 kb. If any SNP in this region has a p-value less than some threshold p0, then cis-methylation QTL is identified. So here we have to use large-scale permutations in the local region for all of the traits to find out the threshold to control the false discovery rate at 5%. So what does the 5% mean? Out of the detected methylation QTLs, we only expect 5% of the CpG sites were detected just by chance. Based on this threshold, we were able to identify over 34,000 cis-methylation QTLs, which accounted for about 10% of the total number of CpG probes after QC. And these cis-methylation QTLs are mapped to over 9,000 genes. As one such example, the strongest cis-methylation QTL in this study is for the CpG probe. For this CpG probe, the p-value equals to 1.5 times 10 to the -73 third. And this SNP explains about 80% of the variance of this CpG probe. Right? This is one such example. But a majority of the methylation QTL have a much weaker effect compared to this strongest one. So here is the distribution of the methylation QTLs across different genomic categories. For the first analysis, you can see that the intergenic region, gene body region, and the transcriptor factor regions accounted for a roughly similar proportion of the detected methylation QTLs. And also, we investigated the OpenC, the CpG island regions and the non-CpG island regions, and these three categories accounted for roughly the same number of detected methylation QTLs. And then we investigated the relationship between the methylational variability and the likelihood of detecting cis-methylation QTLs. So, for example, for each category of the CpG sites, we calculate the proportion of such CpG sites detected with the methylation QTLs. For example, for the category of 5' UTR located in the CpG island regions, we found that only 4.8% of the CpG sites were detected with the methylation QTLs. But for the 5' UTR probes located in the non-CpG ident regions, we detect about 11.5% to be methylation QTLs. So according to this figure, you can see that methylation, the CpG sites located in the CpG ident regions are much less to be detected with methylation QTLs, except for the region 3' UTR. So why this happened? Then, so we found that this phenomena could be well explained by the variance of methylation across the samples. So in this figure, we make a scatter plot. The x-coordinate is the average standard deviation for each category of the CpG sites, and the y-coordinate is the proportion of the CpG sites detected with the methylation QTLs. As you can see that for CpG probes located in the CGI regions, the average standard deviation, the average variance is much lower compared to the non-CGI regions. And we can see that these two measurements are highly correlated. So, and then we will move to the trans-methylation QTL analysis. As I mentioned before, the computational burden for this analysis is tremendous because we have to search all of the SNPs and for all of the CpG sites. And we need to run permutations to generate the non-distribution to be able to control for the false positives. So to do this, we have to first do the quantitative normalization for all of the traits. Because of this procedure, now all of the traits have the same distribution. Therefore, we're only required to run permutations for only one single trait for 1 million times. So this non-distribution can then be used by all of the traits gene-widely. So because of this strategy, we were able to run 1 million permutations for all of the SNPs and then generate this non-distribution. This, based on this procedure, we found that the p-value equals to 2 times 10 to the 10th to control for the false discovery rate at 5%. Okay, so based on this procedure and after several days computation using the NIH BioWolf computational cluster, we were able to identify 585 trans-methylation QTLs. So among those, 438 were discovered to be on different chromosomes. The SNPs and the targeted CpG sites are on different chromosomes. And the rest of 147 were on the same chromosome, but they are 5 megabases away. So before I proceed to do the downstream statistical analysis, we first need to investigate whether the findings based on our study are robust or not. So therefore, we performed a rigorous replication study in the TCGA normal lung tissue samples. Luckily, we have 65 normal tissues based on the TCGA samples. So in these two figures, we plot the effect size for the methylation QTLs in both eagle lung tissue and the TCGA lung tissue sample. As you can see, the directions are largely consistent between these two studies, and the effect sizes are very well correlated. So then we performed a formal replication study. A association was replicated if the replication meets 2 criteria. First, the same direction. Second, it has the FDR less than 0.05. Based on this criteria, we found that overall 65% of our findings could be replicated in independent samples. This probability is not so high because the sample size for the replication sample is quite small. If we are willing to use a more, to replicate the methylation QTLs with the largest effect size, we can see that the replication rate was substantially improved to 93%. Very encouragingly, the trans-methylation QTLs, most of them could be robustly replicated, which is much better compared to eQTL studies. So, very interestingly, we, for the first time, we were able to identify one SNP located on chromosome 16, which can trans-regulate 5 different CpG sites across different chromosomes. These 5 figures show the intensity plot for such association. Interestingly, we found that the target sites are in the CBIIT regions, and all of these associations were replicated in TCGA samples. Among these 5 genes associated with the 5 CpG sites, we found that 2 of them show the classical negative correlation between the methylation and the gene expression levels. So these associations are quite strong, and we identify this potential master regulator using the standard approach. Now we are trying to developing a new statistical methodology to identify master regulators with association not meeting such a single association criteria. So we were able to identify one SNP which is associated with over 60 different CpG sites in trans, and which could also be replicated in independent TCGA samples. This work is still ongoing. So now we are asking a question whether the trans associations could be mediated or explained by the local cis associations. So what does this mean? Let's imagine this scenario. SNP G is cis-regulating a local CpG site, CpG1, and also this SNP can trans-regulate a CpG2 on a different chromosome. Now, by condition on CpG1, if the SNP and the CpG2 are no more correlated, then we say this is a full mediation. So the CpG1 can fully mediate the trans association. So this is a concept borrowed from the causal inference here. If by condition on CpG1, the correlation between the G and the CpG site 2 could be reduced, but it's still significant, statistically significant, then we say the CpG1 partially mediate the trans association. So based on this analysis of framework, we were able to identify, so out of the 585 trans associations, we were able to identify 30 trans associations with full mediation and 166 with a partial mediation. Now, you may wonder what could be the role of these genes for these cis-QTL regions. We identified these genes located in these 196 regions and performed the enrichment analysis. And we found the genes in these regions are strongly enriched with 3 categories: the GTP activities, imprinting, and gene-regulating transcriptions. Yeah. So now we show a very different pattern between the trans association and the cis association. So here, for each category of the genomic categories, we calculate the proportion of the CpG sites detected with either trans associations or the cis associations. As you can see that for cis analysis, the methylation QTLs were strongly enriched with the non-CpG-IDED regions, but for trans associations, such a pattern are reversed. The trans-methylation QTLs are enriched in the CpG-IDED regions. This has not been reported before. as we have demonstrated that majority of the methylation QTLs are not in the CpG island region or gene promoter regions. So, these two regions have been extensively studied because the methylation in these two regions could suppress the gene expression. But given the fact that the majority of our signals are not in these two classical regions, what could be their functional roles? So to answer this question, we have to resort to the public available data for CTCF, histomarks, and DNS hypersensitivity sites in lung-related cell lines. So the question is, are our methylation QTL SNPs enriched in these regulatory regions? So before I proceed for enrichment analysis, we have to define how a SNP, a methylation QTL SNP, is related with a regulatory region, because a SNP could be so given a methylation QTL SNP, if this SNP or its LD SNP is located in the given regulatory region, we say that this methylation QTL SNP is overlapping with a given regulatory region. So based on this criteria, we do the enrichment analysis. So here we use the CTCF binding sites as one such example. First of all of the methylation QTL SNPs, we calculate the lambda 1, which is the proportion of the cis-methylation QTLs related with the CTCF sites. And then we need to generate a control SNP set which has the same number of the SNPs, but then these SNPs are not associated with the DNA methylation. And then we calculate the proportion of the SNPs in the CTCF binding sites. And then we define delta as the lambda 1 divided by lambda 0 as the enrichment score. So when this delta is greater than 1, we say that methylation QTL SNPs are enriched with this CTCF binding sites. So although this procedure is straightforward, during our analysis we found that generating an appropriate set for the control SNPs is quite challenging because of two reasons. First of all, we found that the common SNPs are more likely to be methylation QTL SNPs. Secondly, we found that the SNPs surrounded by many CpG probes are more likely to be methylation QTL SNPs. These two factors, without accounted for, could generate substantial bias or overestimate. So to do this correctly, we need a sampling procedure to generate appropriate control SNP sets. In particular, we need to account for to match the distribution of the minor frequency and also the distribution of the CpG probe density. Yeah. So using the CTCF enrichment analysis as one example, if we do not match for MAF and CpG density, we detect a fold change equals to 4.8. But after rigorously matching for MAF and CpG density, the fold change is dramatically reduced to 3. Therefore, naive procedures may cause overestimate of such enrichment. So based on our procedure, we applied this analysis pipeline to two lung-related cell lines. One is the small airway epithelial cells based on the ENCODE project, which is supposed to be more related with the squamous cell carcinoma. The second cell line is the alveolar epithelial cell lines, which is supposed to be more related with adenocarcinoma. So this data was provided by our collaborator, Professor Ethan Lair's lab at the University of South California. So we tested the enrichment for CTCF binding sites, DNS hypersensitivity, and a few histomarks. Okay, so and this column shows the proportion of the SNPs, methylation QTL SNPs, located or related in these regions for the control data set. And then we calculate the fold change. As you can see, that even for SNPs with only cis effects, all of these categories show enrichment, particularly for the mark H3K4ME3. There's a 6.2 fold change. Now we do this analysis for the trans-only SNPs. We found similar but weaker enrichment. If we are restricting this analysis to the SNPs with both cis and trans effects, such enrichment was stronger. Next, we show that these chromatin markers are increasingly enriched for methylation QTLs with large effects. So we partition the methylation QTLs according to their association p-values, and we see whether if the methylation QTL with a larger effect could have more enrichment for these histone marks, and this is true for this analysis. So, a brief summary for this analysis: we found that both cis and trans methylation QTL SNPs were consistently and strongly enriched for regulatory regions in lung tissue specific cell lines. Therefore, the methylation QTL SNPs may profoundly impact the regulatory function of the lung cells. Now we're going to investigate whether the methylation QTL data are useful for GWAS of lung cancer or other lung-related diseases. So as we know that up to date, the GWAS or GWAS meta-analysis has already identified 5 loci associated with the lung cancer risk for Caucasian populations with the minor allele frequency larger than 5%. These 5 loci are on chromosome 15q and 5p, 6p, 12p, and also 9p regions. Up to date, only one locus located on chromosome 15q, 25. The SNPs were reported to be associated with the total expression of the gene ChRNA5 and also several isoforms in this gene. For the remaining 4 loci, the functional mechanism remains totally unknown. So here we are going to investigate whether these risk SNPs affecting DNA methylation in a local region or in a distal region to increase the risk of developing lung cancer. So first of all, I would like to use one example located on RAD52 to explain the potential of this data set. So the SNP located in RAD52 region was first discovered by us and was published 2 years ago in Cancer Discovery. But this SNP showed no signal in the eQTL analysis. So to investigating the hypothesis of DNA methylation, we checked the association for all of the CpG probes located in this local region and the SNPs and make this association plot. So each plot here, each point represents the log p-value for a given CpG site. As you can see, we can identify one CpG probe located in the gene body of the RAD52, which is strongly associated with the given risk SNP. And we found that such association could be robustly replicated in independent TCGA samples. So although we cannot for sure say that the DNA methylation is involved for the lung cancer carcinogenesis, but this analysis shows the potential of use the GWAS of use our methylation QTL data to interpret the existing GWAS signals. Okay, so encouraged by this finding, we extended this analysis for all of the GWAS loci for lung cancer. First of all, we found that for the most important locus, 15q25, there are 2 independent SNPs contributed to the risk of lung cancer. We could show that both of the SNPs regulate CpG sites in the local region. right? And then we investigate the signal for 5P third region and the MHC regions and RAD52, and also for CDKN2A. So out of these 7 SNPs located in 5 genomic loci, 6 of them show very strong methylation QTL signals, and all of these could be strongly replicated in TCGA samples. The only exception is for the SNP located in the NIP21 region. In this region, we could not identify the strong signal for methylation QTL. However, given the fact that the CpG density is very low in this region, we could have already missed the information here. So this data suggests that there's a great potential to use the methylation QTL data to interpret existing GWAS signals. Here, I'm going to provide another important, informative example to explain the usefulness. So this SNP, this is a very complex genomic region, the MHC. This region has been well known because of its extremely complicated LD pattern, because the SNPs located in this genomic region have very long-range LD patterns. So it's very difficult to nominate the genes relevant to the disease. So originally, a SNP RS3117582 located in BAT3 was reported in Nature Genetics as the lung cancer disease gene. Okay? But then we found that this SNP and another SNP located in MSH5, these two SNPs are in perfect LD. So without any external information, we cannot differentiate which gene could be more likely to be the disease gene. So based on our methylation data, we can see that the SNPs regulate, or it's strongly associated with a CpG site located in the MSH5 region instead of the BAT3 region. So this analysis, provide evidence that MSH5 instead of BAT3 could be the relative gene for the lung cancer risk. And more interestingly, we also found that this SNP trans-regulate a CpG site located on the gene FBRS1L1, which is located on chromosome 12. So this phenomenon also raises the possibility that this SNP trans-regulate a distal CpG site to affecting lung cancer risk. So although we could not provide conclusive evidence, but our data is very helpful to generate very important scientific hypotheses for future investigations. The natural next question is how could we use the methylation QTL data to prioritize the SNPs in the future GWAS studies or kinase gene studies? So to answer this question, we must show that the methylation QTL SNPs are enriched compared to the rest of the SNPs for lung cancer risk. To do this analysis, we first have to do some intuitive analysis. So we use the NCI GWAS of lung squamous cell carcinoma with over 5,000 cases and 1,400 cases to make this quantile plot. To do this analysis, we first have to prune the SNPs so that no pair of the SNPs have correlation R² larger than 0.1. In this case, we can reduce the complexity of the correlation. And second, because our goal is to investigate the potential for unknown genomic regions, we exclude all of the we have excluded the regions with established GWAS signals. So then we make this QQ plot. So as you can see, the x coordinate is the log p-values for this GWAS for all of these SNPs, and then the y coordinate shows the log p-values for the methylation QTL SNPs. So here I do analysis for 3 categories for SNP methylation QTL SNP mapping to the North Shore regions or the gene body regions, or for SNPs mapping to the CTCF binding sites. As you can see that all of these 3 Q plots deviates from the expected 45-degree line, which clearly demonstrate that there are some enrichment for this methylation QTL SNPs. Okay, so but the formal enrichment have to be tested by a formal testing procedure. So we developed a testing procedure for testing the enrichment using the whole NCI lung cancer GWAS data of European ancestry, which included about 6,000 cases, 6,000 controls with 3 subtypes: squamous cell carcinoma, small cell, and adenocarcinoma. And we have established 5 GWAS regions with the focus on unknown regions. So here's our procedure for testing for association. First, given a set of K-SNPs with association p-value P1 to pK, we test whether they follow a uniform distribution. Because if there's no association, then these p-values will follow a normal distribution. And then we transfer the p-values to the chi-square quantile, and then we perform a test that uses this statistics with f set to 0.1. So as we showed previously that the specification of f does not affect the sensitivity of the test. So it works best if we know the proportion of SNPs associated with lung cancer. But because this cannot be known in advance, we have to specify this parameter here. Yeah. So then we perform 10,000 permutations to evaluate the significance. Okay. Based on this analysis, we can show that if we include all of the methylation QTL SNPs, we found that these SNPs are strongly enriched in the overall lung cancer risk after excluding the GWAS signals. And also we can show that this enrichment was primarily driven by the association in the squamous cell carcinoma. Yeah. So if we do the analysis by stratifying into different categories, then we can show that such enrichment was primarily driven by the signals in the North Shore region. Very interestingly, the methylation QTLs mapping to CpG island regions show very little evidence for such enrichment. Given the fact that we have traditional epigenetics studies have primarily focused on gene promoter region in the CpG island regions, here these findings suggest that we need to treat other regions equally well. And we also perform analysis for different regions in terms of the gene locations, and we found that methylation QTLs located mapping to the promoter regions show very little enrichment, but the methylation QTLs mapping to the gene body regions show very strong enrichment. Yeah. And further analysis will show that this the signals were primarily driven by the SNPs mapping to CTCF binding sites and the H3K27ME3 histomarkers. So these analyses show that the methylation QTL SNPs mapping to non-promoter gene region and the non-CpG island regions show very strong evidence of enrichment for lung cancer risk. For the traditionally important regions, our data do not support such enrichment. So what could be a possible explanation for this possible carcinogenesis? A paper published 25 years ago by Peter Jones' group, they showed that the gene body and the non-CpG island regions are more likely to be mutated when they are methylated. So when the methylation QTL located in the gene body and the non-CpG island regions, it will create a lot of variation for the DNA methylation in these regions. So therefore they will, create the increased risk for these regions. So these results suggest that we need to put a similar emphasis on these untraditional regions for the genetic analysis. So as we know that the methylations are very tissue specific, so the natural question is that whether the genetic control of DNA methylation could also be tissue specific. To answer this question, we have do the analysis in independent TCGA breast and kidney tissue samples. We try to replicate all of the cis and trans associations in these two data sets, and we found again that between breast and lung, between kidney and lung, the effect size across these two studies are highly correlated. So based on a formal replication study, we found that overall for breast, 55% of cis could be replicated, and for kidney, 70% could be replicated. The difference between these two replicates could be attributed to the different sample size between these two studies. Importantly, if we raise the bar for the cis association to try to replicate these stronger cis associations, we found that the replication rate could be substantially increased. And very encouragingly, we found that majority of the trans associations could be replicated, which is very different from existing expression QTL studies. For example, in a recent paper for mapping expression QTLs based on adipose, LCL, and skin tissue samples, they found that for the trans-meQTL signals, only about 50% could be replicated across different tissues. Our study shows that the genetic control of DNA methylation shows little level of heterogeneity across different tissues, which is important because we can use this information to borrow strengths across different tissues to increase the power to detect the methylation QTLs if we were able to combine different tissue samples. So here's the brief summary of this talk. First of all, genetic variation profoundly and extensively impacts DNA methylation in the lung tissue. And we found that the cis and trans methylation QTLs show distinct patterns in terms of the proportion of detected methylation QTLs. And we, for the first time, proved the existence of a master regulator exists for DNA methylation. And we show that the genomic control of DNA methylation show modest heterogeneity across different tissues for both cis and trans. And we also show that the methylation QTL SNPs may profoundly impact the regulatory function of lung cells. In terms of the genome-wide association study for the lung, we show our data could be very useful for interpreting the existing GWAS signals, for prioritizing SNPs for future studies, and also for improve the polygenic risk prediction, which is our current project. So I want to highlight that the traditional methylation study have focused on gene promoter region and CBIIT region because there are classical negative correlation between expression and the DNA methylation. But our data here highlight the link between the lung cancer carcinogenesis and the non-promoter, non-CBIIT regions. So our methylation QTL data with the 1,000 genome imputation now are available to download from this FTP server, so with the help from Carl McCabe in our division. In fact, this study has been performed under extensive collaboration with many collaborators from DCEG, University of Milan, University of South California, University of Chicago, and the Broad Institute. I'm grateful to all of my collaborators, and thanks for coming.
Thank you, Dr. Shi, for a very interesting presentation on this important work. So I'll open the floor for questions, and here in the room, just turn on the microphone on the desk in front of you, and on the phone, just raise your hand on the WebEx, and we will get to you. Max, did you have a question?
Oh, yeah, I have, I guess, several questions, if I may ask some. So one question I have, I think it's a very interesting observation you have in terms of the difference between CpG iron versus non-CpG iron in terms of like the trans, the MQTL association with the CpG iron, and the cis actually are associated with non-CpG iron. Now, one thing has been known very well, like in the methylation field, are the CpG iron actually typically are not really methylated in normal like tissue, right? That's probably one of the reasons actually, like if we especially see the distribution, you get more like cis instead of the trans. So I was just wondering, for example, like in your case, like all those the trans, like the target, and what do you know about those genes? Because typically the genes are known are either like X-linked genes or like imprint gene in terms of like being methylated in the like a CpG target, but which is very actually rarely actually like show any kind of methylation like at all variation. So I was just wondering whether you have any like study and comment.
This is a very good question and a very difficult question. So we were also confused and intrigued by this question for a long time. In fact, we have tried different kind of analysis, including, we try to use the high-seq data to map the distal interaction between distal genome regions, but none of our analysis have produced the interesting or the positive findings. So in terms of your question, so my answer is we still don't know. But any discussion will be welcome.
Just maybe a follow-up with the second question in terms of the cis. So when you're actually looking at all those cis, do you really see some kind of pattern in terms of the distance among the polymorphic site?
Yes.
So what kind of pattern you can tell us about?
Well, so majority of the SNPs is about 10 to 20 kb related to the targeted CpG site. Yeah.
And what would happen, because typically the HMH 450 density is quite high. You actually would have a cast-off for those like methylation target site. So do you see some like, are they usually like correlated in some way, or you actually see these things scattered around? You see what I mean? Because you have like a cast of all those target sites. And do you see they always show some kind of like cis-meQTL, or not really necessarily? They could be like
thank you for reminding us. So I think this will be a very interesting analysis. We have not tried this before. Yeah.
Actually, just have a sort of a philosophical question. As you mentioned before, that traditionally the methylation is controlling the expression of the gene.
Right.
It's usually in the promoter. So when you're looking at some of the analysis that you showed that is in the body of the gene, what would be the mechanism or the function? What would be, because in general we assumed that it was loss of the expression. So how does one? Do you have any idea of what might be doing there? I mean, like, what is the, what's the significance of having
Well,
methylation site on the body of the gene rather than the promoter?
There are some papers show that the DNA methylation in the gene body region could affect the splicing and also, the gene inauguration. But I don't know if there are some, exact biological mechanism. But from the analytical point, I don't care much about this, because if we are interested in use the data for the future prediction or the prioritization of the SNPs, the findings can be used without considering the functional mechanism.
Yeah. No, without a doubt, this is finding. Did you ever look at the expression level in comparison to some of these? You showed 5 genes there that were very
Right.
closely associated, with some of the methylation.
Right.
But I was wondering if you had actually looked
Yeah, so because the TCGA have only very limited number of RNA samples for lung tissue samples, so we were only able to investigate the association for a few genes. Yeah. Okay.
Can I maybe ask a couple of more questions here, if you don't mind? So I think it's very interesting when you comment the actual difference between the eQTL and the mQTL, because eQTL actually very much known to be dependent on the different tissue, because we know actually the importance of the gene expression in terms of tissue-specific expression pattern. However, what you see for the mQTL, they seem to be really less dependent, because you can replicate these things in kidney and breast tissue, and similar to the ego. So do you think here they are more like a constitutional in some way because they are determined by the sequence, underlying sequence? Is that what you're thinking, or?
Not yet.
Okay.
Not yet.
I mean, also maybe something related to this. If it's really determined by sequence, is that really like a local sequence context? Or maybe actually something further, like involve like a long range of interaction? So that's how things related even for the cis, because you talk about something that's 10 to 20 kb away. And there are also many other data, for example, all those like complete conformational cage, capture using like C3, C4, C5 data. So does that actually help you in terms of interpret or understand those interactions there?
Well, we don't know. So as I said, we tried this analysis to use the 5C or high C, this kind of data, but we did not produce interesting findings.
If I may just comment on the mutation relationship between methylation and so for example, like polymorphism, they are also very common, like for the CT. And part of the reason is also has to do with because we now talk about like evolution. We talk about population. So in that sense, because many of those things are methylated, that's leading into the mutation. And which actually not just like occurring in the germline, but also, for example, happening at the somatic mechanism of somatic mutation as well. All those things are kind of consistent with what the study you present.
That'd be great. Yeah. I'm happy to talk.
You showed a very interesting example of a correlation of the methylation data with GWAS prediction of susceptibility gene. I was wondering in how many other cases have you seen that strong a correlation?
Well, here we only investigated the lung cancer GWAS. So our current project with Dr. Taranani here, so we are trying to use the TCGA data to systematically mapping the methylation QTL across different tissues. And then we will be able to systematically, to investigate the enrichment across different cancer GWASs. I think that study, when done, will provide more information and insight.
Yeah. In the GWAS study, you have control, you have a case. But in this MEQTL, you only use the normal tissue from cases, right?
Right, the adjacent.
Adjacent. So there's no control. I mean that normal control.
Practically, this will be very difficult because healthy people are unlikely to contribute to the tissue samples.
Okay. Yeah. Another question. So the tissue-specific methylation pattern. Do you show there's a comparable pattern between breast and the kidney and the lung? Have you looked into the difference? I mean, the methylation patterns are very different. Tissue-specific pattern. We know the general knowledge. Did you look into this? There's some difference between breast and lung in terms of MEQTL?
Yes. In fact, this is the ongoing project. We are trying to do the functional enrichment analysis for the methylation QTLs showing strong heterogeneity across tissues. Yeah.
the follow Tony's question. I mean, I found that initially mentioned about 34,000 probes and 9,000 genes that this relationship. So GWAS standard is one way to narrow down. So I just wondering if there any other way you have to narrow down just a few like genes that have more dramatic effect on the lung cancer.
Could you please repeat your question?
Yeah, right now you identify the solution to the 34,000 probes and 9,000 genes. So I was wondering if there are other ways to narrow down this number of genes which has a big impact on the cause of lung cancer.
Well, I don't know. I don't know your exact question. So
So can you narrow down the number of SNPs?
Yeah, number of genes. Basically, right now I feel like it's identified 9,000 genes. That's a lot of genes have impact on the lung cancer. So if you have other data you can use or other method you can use. Yeah.
Oh, you are talking about our enrichment results, right? Well, I think this is a good question. So for these strongest signal show in this QQ plot, we actually trying to replicate this single findings based on larger meta-analysis. But honestly, we could not replicate these top signals. Yeah. But we acknowledge that such kind of enrichment pattern is still there. So currently, my branch chief, Dr. Jianxin Shi, has been collaborating with me to use such a enrichment signal to improve the polygenic risk prediction. Yeah. So whether these top findings could be further replicated in a bigger sample size, we cannot predict based on the current knowledge.
So I have a few comments also, since we are working together and maybe, more on this type of question. So first of all, for the top SNPs here, we inserted these top SNPs that are 20, really, really high, in a consortium-based analysis, the OncoArray analysis in this consortium that includes analysis of different type of cancers with hundreds of thousands of individuals. And we are expecting the results of this replication very soon. So we hope that we will be able to confirm this in much larger sample size. In terms of the questions about the normal tissue, that of course, as Jianxin said, it's practically impossible to obtain, not diseased lung. We contacted and collaborated with the investigator in USC precisely to obtain purified normal lung cells that we obtained from autopsy, from a car accident, from normal people that had accident. And so they were able to cultivate the cells from type 1 to type 2 cells. And then we replicated this analysis based on these cells precisely to have as close as possible to a normal tissue. And this was done and confirmed at USC. Another question that Dowd raised on the point of the function, what is, because from our point of view being, medical doctor, we care for that question. And the paper that Jianxin mentioned already from many years ago, published in Science from the group of Peter Jones, and it was confirmed later on in many other papers, we believe that it could be important because in the body of the genes, so there are highly methylated areas, as we have seen. So there is this high possibility that the demethylation of this area will then produce, instead of the uracil, a thymine, and thymine is less recognized by the DNA repair, and this will increase the risk of carcinogenesis without affecting the gene expression. TP53, for example, is highly affected in this way, and the recent TCGA analysis of tumors across different types, published by Lawrence et al. In Nature, show that there are type of mutation that could be related to this mechanism, and that actually they are the large majority across different tumors. So this effect could be really important.
Great, thank you. I'm afraid we're out of time. I want to thank everyone for the excellent questions and comments. Hope you can all join us for our next presentation, which will be on October 1st, when Dr. Ashley Wilder-Smith from NCI's Outcomes Research Branch will present on a person-centered assessment resource, an innovative health measurement system. Once again, thanks to everybody who's joined us today, and let's thank our speaker, Dr. Shi, one more time.